I'm Katie and welcome to my channel Biology by Katie. If you have watched my previous videos, you will know that I am making my way through the A-level AQA biology syllabus, topic by topic. If you haven't watched any of the previous videos, then that's what I'm doing. I hope you find the videos helpful, useful and easy to follow along with. And if you do have any recommendations or requests, please do let me know in the comments. We're on to topic 1.3 now, titled lipids. So lipids are another one of our biological molecules, one of the 4 organic polymers that make up the biochemical basis of life. Lipids are quite a large group of molecules, but they do all share some essential properties. So first of all, they are made up of carbon, hydrogen and oxygen molecules only. This is very similar to that of carbohydrates, but lipids have a lot smaller amount of oxygen. Next, they are insoluble in water, meaning that they will not dissolve in water. However, they are soluble in organic solvents, so these are carbon-based substances such as alcohols or acetones. There are 2 main types of lipids that we are going to look at in some detail. The first are triglycerides and the second are phospholipids. Starting with triglycerides first. So on the left here is the polymer form of the triglyceride. So we have all of the subunits bonded together. The term triglyceride can be broken up to give us an indication of what we're looking at here. Tri is the Greek prefix meaning 3, whilst glyceride refers to a molecule that is formed when a glycerol bonds with fatty acids. So on the right, we have the monomer units, 1 molecule of glycerol and 3 separate fatty acid chains. So here is both the molecular and the structural formula of glycerol, a very simple polyol molecule. Glycerol specifically is a triol as it contains 3 hydroxyl groups, which we can see there on the right-hand side of the molecule. Then we have the 3 fatty acids. Now, there are hundreds of different types of fatty acids. The important thing that we recognise is that they are hydrocarbon chains with a terminal carboxyl group. Therefore, they are carboxylic acids. So they have that COOH group there at the end. This is important to remember as it is the carboxylic group that interacts with the glycerol in order to form these new bonds. Because there are so many different variations of the fatty acids, we use that R to represent the variable hydrocarbon chain component in order to keep these diagrams neat and make sure that they're not confusing. The fatty acid chains themselves are classified further as either saturated or unsaturated. So here on the screen is an example of a saturated hydrocarbon chain. What this means is that each carbon that is not involved in the carboxylic group forms only single bonds to other carbons, and they have as many hydrogen atoms bonded to them as possible. So we can see there are no double bonds except for the carbon that is involved in the carboxyl group. Unsaturated fatty acid chains, however, contain at least 1 carbon-carbon double bond. So here we have a monounsaturated fatty acid. So what that means is we have just the 1 carbon-carbon double bond there. Remembering mono means 1. So monounsaturated 1 carbon-carbon double bond. Or we can have polyunsaturated fatty acids where there is 2 or more double bonds. So poly meaning many or multiple. As it's stated at the top there, building triglycerides from their monomer units is a condensation reaction. So therefore, a single molecule of water will be released per new bond formed. When we line the glycerol and fatty acids up next to each other, we can identify where this reaction will occur. So where hydrogen times 2 and 1 oxygen will be removed from, and therefore where the new bonds are going to form. So at the top of the screen here, we can see we have our monomer units pre-reaction and we've identified where the 3 water molecules will be removed from. At the bottom, we have our resultant polymer, in this case, our triglyceride. So that's 3 fatty acids joined to a single glycerol molecule. The bond formed when lipids are built is called an ester bond. So in this case, we will have 3 ester bonds where each of the carboxylic acids or fatty acids has been joined to the glycerol. Right, let's move on to look at the second group of lipids, our phospholipids. So phospholipids look quite similar to triglycerides at first glance. The difference between the two is the presence of a phosphate group in the place of a third fatty acid. So whereas triglycerides have one glycerol and 3 fatty acids, phospholipids have one glycerol, 2 fatty acids and a phosphate group. We have a very similar pre-reaction arrangement of the molecules here. We know that the carboxyl groups of our 2 fatty acids will react with the hydroxyl group on the glycerol. That does not change. In terms of the phosphate group is the result of bonding with a number of different phosphate-containing molecules. This here is dihydrogen phosphate, but all we need to know is that the addition of the phosphate group is also a condensation reaction, and therefore a water molecule will be released. So we still have the release of those 3 water molecules, and we still have 3 new bonds being formed. So let's look at what our final phospholipid looks like. The central glycerol molecule, 2 fatty acids joined to the glycerol via an ester bond, 1 ester bond each. The phosphate is also joined by an ester bond, but we call it a phosphate ester bond for the obvious reason that we have a phosphate group. It is important to always show the bonding of the phosphate and the fatty acids on opposite sides of the glycerol molecule. So we can see there the fatty acids are on one side and the phosphate on the other, as this structure is very, very important to their later behavioural properties. Due to the overall negative charge of the phosphate group, it is attracted to water, so therefore we describe it as being hydrophilic. The fatty acid chains, however, have non-polar bonds throughout, which will not interact with water, so therefore they are described as being hydrophobic. So there we have our final phospholipid molecule. Moving back to triglycerides for a minute to discuss their functions. Firstly, they are an extremely valuable long-term energy source. They have a lot of carbon-hydrogen bonds. Now, these carbon-hydrogen bonds hold a lot of energy, so it's very useful if energy is needed and polysaccharide levels are low. Second of all, waterproofing. We mentioned that the fatty acid components are non-polar and therefore hydrophobic and thus waterproof. A good example of this in action is the waxy cuticle produced by plants to reduce water loss at the leaves. Organ protection. There is often a layer of fat around major organs to act as shock absorbent and to protect the organs from damage or harm. And finally, insulation. So fats do not let thermal energy pass through easily. They therefore can help to maintain homeostatic environment by resisting extreme heat changes. Next, let's have a look at the functions of our second group of lipids, the phospholipids. Now, because phospholipids are quite structurally similar to triglycerides, they can do a lot of the same things. So they're used for energy storage, waterproofing, insulation, and protection. But they do, however, have an added function, and that is being the major component of cell surface membranes. So let's have a look at why their slight difference in structure means that they can form this plasma membrane. So as we mentioned, a phospholipid is a polar molecule. It has sections within a single molecule that both look and behave very differently. The phosphate group is negatively charged and therefore hydrophilic. The fatty acid tails are non-polar and therefore they are hydrophobic. We know that the internal environment of all living things contains water. In humans, for example, connective tissue is aqueous, extracellular fluid is aqueous, cellular cytoplasm is aqueous. So here we have the hydrophilic heads interacting with the aqueous environment, so with the hydrophobic tails facing away. But the problem is we have water-based substance on the other side too. So the phospholipids arrange themselves again so that the hydrophilic head interacts with the water and the hydrophobic tails are facing away. So in the end, we end up with this double layer of phospholipids that are facing towards each other. This therefore protects the hydrophobic tails from having to interact with water by shielding them within the arrangement. This structure is therefore called the lipid bilayer, with bi meaning two, and the lipid layer obviously meaning the layer of phospholipids. So let's check through the AQA specification points and make sure that we have learned everything that we need to know. So the first point is, Triglycerides and phospholipids are two groups of lipids, so we identified those right at the start. Secondly, triglycerides are formed by the condensation of one molecule of glycerol and 3 molecules of fatty acids. So when we looked originally at the formation of a triglyceride, we split it into its monomer units and showed that we had those 4 monomer units. Next, a condensation reaction between glycerol and a fatty acid forms an ester bond. So remember, we had the removal of a water molecule, and round that remaining oxygen atom is the new ester bond. Next, the R group of a fatty acid may be saturated or unsaturated. So saturated fatty acids were where there was no carbon-carbon double bonds and as many hydrogens bonded to the carbons as possible. And unsaturated was where we had the presence of a carbon-carbon double bond, whether it be mono or poly unsaturated. Next one, in phospholipids, one of the fatty acids of a triglyceride is substituted by a phosphate-containing group. So there we had the 2 fatty acids on the right-hand side and the phosphate group on the left, which gave the molecule its polarity because we had the negatively charged phosphate ion and we had the hydrocarbon change of the fatty acids. Next, the different properties of triglycerides and phospholipids related to their different structures. So we know that because they were quite structurally similar, they did do some very similar functions. So we had energy storage, waterproofing, insulation and protection. But with the phospholipids, we had that added function of being the main component of a plasma membrane. So in terms of the AQA specification points, we have learnt all of those, so that is topic 1.3 complete.